In 1895, Boder discovered complement. Complement is actually the system of plasma protein which is activated directly by pathogens or indirectly by pathogen bounded antibody. Okay, so let me explain you. When any pathogen or bacteria attacks us, when the pathogen attacks, then the antibodies in our system attach to the antigen of that organism or pathogen. When antibody attaches to the antigen of pathogen, then it activates. When antibody activates, it also activates our complement system. This is how our complement system activated indirectly. In 1899, Beijing proves tobacco mosaic virus, which causes tobacco mosaic disease. It's a plant disease which damages the leaves of plants. Armengam discovers Clostridium botulinum in 1896. In 1900, Reed proves yellow fever transmitted by mosquito. In 1902, Landsteiner discovers blood group. In the year of 1910, records show Rocky Mountain spotted fever caused by microbes. Rocky Mountain spotted fever, commonly known RMSF, is a bacterial disease spread through a bite of an infected tick. Ticks are small spider-like creatures which feed on the blood of birds and mammals, including humans. In 1903, Wright and others discovered antibodies. In 1906, Weizmann developed complement fixation test for syphilis. A complement fixation test, or CFT, is a classical laboratory diagnostic test which is used for determination of virus antibodies in patient sera or cerebrospinal fluid samples during an acute infection. Rose discovers a virus can cause cancer in 1911. From 1915 to 1917, De Herelle and Thor discovered bacterial disease. In 1921, Fleming discovers lysozyme. He also discovered penicillin in 1929. Lysozymes are naturally occurring enzymes that play an important role in the innate immunity, providing protection against bacteria, viruses, and fungi. They are protein present in many mucosal secretions like tears, saliva, and mucus. And penicillin is an antibiotic used to treat infections caused by bacteria. It doesn't work on viral infections such as cold and flu. Alright. In 1923, first editions of Burger's manual occurs. In 1928, Griff discovered bacterial transformation. In 1935, Dumac. It's Dumac or Dumac. Anyways, is it? Okay. In 1935, Dumac discovered sulfur drugs. In 1933, Ruska developed electron microscope. It's a very important man in microbiology. Okay. In 1931, Van Yale studied photosynthetic bacteria. In 1941, Beadle and Tatum proposed one gene, one enzyme theory. It suggests a single gene produces a single enzyme. Oh, that's great. Okay, in 1944, Waxman discovered streptomycin. In 1937, Charlton divided living organisms into prokaryotes and eukaryotes. In 1949, Anders, Weller, and Robbins grew poliovirus in human tissue culture. Weston and Crick in 1953 proposed DNA double helix structure. In 1961, Jacob and Monod proposed lake operon. Lake operon or lactose operon is a series of three genes in bacteria that produce the necessary enzyme to obtain energy from lactose. In 1959, Yalo developed radioimmunoassay. The radioimmunoassays are used for detecting the concentration of a specific antigen or substrate in sample using antibodies. The enzyme restriction endonucleases was discovered by Arber and Smith in 1970. Actually, endonucleases are bacterial enzyme that cleaves double-stranded DNA. Woese divides prokaryotes into bacteria and archaea in 1977. Genentech and Lysand in 1980 developed scanning tunning microscope. In 1982, recombinant hepatitis B vaccine was developed by Maurice Hellman and his team. From 1983 to 1984, Gallo and Montagnier identify and isolate HIV.